In today's video, we're going to talk about this protein right here, Ras-GTP. You might be forced to study this protein by your professor. You might be interested in it yourself for research purposes. When I started first learning about Ras, I was questioning why we were focusing on this protein, which is involved in cell signaling pathways, when there's usually tons of proteins involved in cell signaling pathways. You can think of Ras as kind of an intermediary protein. So it's going to receive signals from usually transmembrane proteins attached to the plasma membrane. Ras itself is attached to the plasma membrane, but it doesn't extend beyond the plasma membrane. So if this is the membrane right here, Ras is going to sit on the inside part of the membrane, so it doesn't have a part that's going to extend this way towards the cytosol. So that's why it needs the help of transmembrane proteins, such as in this video we'll focus on RTK, receptor tyrosine kinases. So receptors like these actually extend beyond the membrane, so they can bind to different ligands that come along in the extracellular space, and that's why Ras functions as a kind of intermediary between receptors and transmembrane proteins here, and Ras will also relay the signal that it gets from these transmembrane proteins to other proteins. So it's a good habit to think of Ras as an intermediary protein, and its main function is going to be in cell division. So that's why your professor might be highlighting Ras a lot, or why your textbook might be talking about it. Because when Ras goes awry, when it's always activated, it's going to be always telling the cell to divide in downstream signaling. So this is really important in deleterious mutations or in conditions like cancer. So that's why we're focusing on Ras today. So like I said earlier, Ras sits on the inside of the cell membrane. So it's going to sit somewhere here, attached to the inner leaflet of the membrane. If we zoom into this here, we can view Ras in more detail. So Ras in its inactivated state is attached to GDP here, and it's going to sit on the membrane, and it's going to have this tail that is a hydrophobic tail that's going to extend into the hydrophobic parts of the phospholipid bilayer. It also has other biochemical properties that allow it to bond very well to this membrane. So what you need to remember is that Ras has a tail that's going to help anchor it to the inner part of the lipid membrane. Again, anchor to the inner part of the lipid membrane. Right next to Ras, we're going to imagine, is the RTK. The full name is receptor tyrosine kinase. It's a type of protein receptor, but we're just going to call it RTK for the simplicity of not having to say all that. RTK is right here. So now that we know about the placement of Ras inside the cell, let's talk about its structure. So the Ras protein is around 188 to 189 amino acids long, and somewhere around the 100 amino acid mark, it has a binding site for this molecule here called GTP. For those of you that don't know, GTP stands for guanosine triphosphate. Guanosine, to help you remember, guanosine is a word that describes a guanine base bonded to a ribose sugar. So whenever you hear guanosine, think guanine and ribose. Ribose sugar is here. It's a 5-carbon ring. We can count 1, 2, 3, 4, 5 carbons. And right here is our guanine base. Guanine is, of course, one of the 4 bases involved in DNA. It's one of the 4 nitrogenous bases you'll find in biology very commonly. So our guanine is here, our ribose is here, and right next to it you'll notice that there are 1, 2, 3 phosphate groups. Each of these are phosphate groups. You can see because of the phosphorus atom in the middle. That can help you remember it. So guanosine triphosphate intuitively has 3 of these phosphate groups. If we are to remove one of these phosphate groups, we get guanosine diphosphate. I hope that's also pretty intuitive coming across. So guanosine diphosphate is what Ras is initially attached to. In its inactive state, Ras, as you can see by this tag here, is attached to GDP, guanosine diphosphate. When Ras is in its active state, this guanosine diphosphate will be replaced with a guanosine triphosphate. So you can think of this as a kind of go signal for the molecule, whereas guanosine diphosphate is kind of a stop signal for the molecule. So let's focus now on how, in our case, receptor tyrosine kinases are going to affect this RAS molecule. So we have our receptor here, and we know that this part of the receptor is going to bind to a ligand. So in the case of receptor tyrosine kinases, ligands are going to come from the extracellular space, and they're going to bind to these pockets of the RTK. Once that happens, RTKs are going to bind together. We're going to call it dimerization. Two RTKs are going to come towards each other, and then they are going to phosphorylate each other, which means they're going to add phosphate groups to their tails here. So once that happens, it means the RTKs are kind of in their active state, because different proteins are going to be attracted to these new phosphate groups. So different proteins are attracted to RTKs depending on the signal. In the case of RAS, we're going to consider a couple. So usually what happens is a SHC protein might bind to the RTK. Sometimes a GRB2 protein binds directly to the RTKs. So either the SHC is going to bind first, and then a GRB2 is going to bind to the SHC molecule, or the GRB2 is just going to bind directly. Either way, in either of these scenarios, a GEF protein is going to be recruited. So now a GEF protein is going to recognize the GRB2. And the GEF protein is what we're going to care about in this video, because the GEF protein, which stands for guanine nucleotide exchange factor, is going to find free-floating GTPs in the cytosol, which there are many. The cell loves to manufacture GTPs. It's going to grab that GTP, and it's going to replace the GTP for GDP on RAS. So now this RAS protein here, which previously had GDP, is going to be replaced with GTP protein. That is going to activate the RAS protein. From there, RAS is going to do all sorts of stuff. We're going to get into that in the later parts of this video series. But for now, what we need to remember, RAS is a protein that lies on the inner leaflet of the membrane. Its function is tightly correlated with GTP and GDP, two different versions of guanine molecules. It interacts very closely with receptor tyrosine kinase and other receptors on the membrane, and it's important for cell division and signaling. In the next video, we're going to get into GAP, which helps convert GTP back into GDP, and we're going to get into how RAS is mobile throughout the cell and its other cell signaling properties.